Hi, everybody. Uh, Welcome to the webinar. Thank you guys for joining us today. I hope you and your loved ones are keeping well in this time of crisis. So I'm Soumya Luthra. I'm a research lead at Elucidata, and I would just like to quickly set the stage for the webinar today and introduce the presenters. I hope you guys can hear me fine. So before we get started, let me just give you a brief outline of the session. So this webinar is being recorded, and a copy of this webinar will be shared with you over email. And we would be taking all the questions in the last 10 minutes of the webinar. And we also have a team of application science scientists. And if you have any doubts or questions during the demo, please feel free to post them on the chat and we will reply promptly. All right, so let's get started. So just to take you through the agenda, so in today's session, we'll essentially be talking about single-cell omics data, a technology which in the past decade has revolutionized our understanding of normal cell types, biological functions, and disease biology. So we'll be taking you through an exciting story of how we are able to derive novel biological insights into embryonic neural development using a publicly available single-cell data set. And we'll follow that up by talking more about how we can enable you scientists to use single-cell data more effectively with our platform and services. And towards the end, we will share with you a product video to illustrate some of the single-cell capabilities in action. And we'll end that with a Q&A session towards the end. So great. So as we get started, let me just quickly introduce ourselves at Elucidata. We are a young team. We're based out of the US and New Delhi in India. And our mission as a company is to enable scientists to use biological data more effectively for their research. And we do that using our platform and our services. And in the past 4 years, we've built a very strong cross-functional team of about over 60 people, and we've driven impact with over 40 partners across academia and industry. So without much further ado, I would like to welcome and introduce our presenters for today. So today we have with us Dr. Sidhartha Goyal. Sidharth is an assistant professor in the Department of Physics at University of Toronto. And we've collaborated with Sidharth's lab over the past few years, working together to unravel the metabolic landscape of neural development. And I will let him comment on how physics led to his interest in embryonic development. And we also have today with us Abhishek, Dr. Abhishek Jha. Abhishek is the co-founder of Elucidata. And in addition to his experience at MIT and University of Chicago, he has over 5 years of experience at Agios Pharmaceutical, where he was a key driver of 2 different FDA-approved drug discovery programs. And with that, I would like to hand it over to you, Sidharth.
I guess I'll start by answering. Can everybody hear me? Good. So I'll start by answering Soumya's question about how physics led to neural development, and the short answer is I think most scientists and physicists, I think in general, are very opportunists, and biology being an exciting area and having sort of scientists working in the neighborhood. So this neural development project, although we worked on data which is publicly available, it was actually generated by a colleague at University of Toronto, which was exciting, and that was sort of partly why embryonic neural development. But the exciting part was also developing a technique how to use single-cell transcriptomics to sort of do more general things beyond neural development. Okay, so let me sort of take you through a few of our slides, a project which we have recently worked on to leverage single-cell transcriptomics to understand embryonic neural development, and this is in mice. Okay, so metabolism in general is sort of well understood very broadly across all life forms. And in particular, it has sort of also not only linked with other biological processes, but also with cellular differentiation and function. And by cellular differentiation, the idea is that, of course, all multicellular life starts with a single cell called a zygote, and it takes various divisions, and we all know this caricature of as these cells divide, they change the character from what they started from, and it better do so we can actually have an eye to see and the liver to metabolize our alcohol. So during this process, which is called cellular differentiation, it turns out that metabolism plays a fundamental role, in the sense that metabolism changes drastically as these cells progress from an early state to a later state in differentiation. Um, and the challenge now, or a more recent challenge slash opportunity it has provided, is that perhaps one can also tweak cellular differentiation by knowing enough about metabolism and metabolites and underlying drivers of metabolic reactions. So let's go to the next slide. So here, so with this general introduction, let me sort of introduce the single cell data we use for this problem. So on the right, there is a schematic of the paper from which we started, which generated the first round of this data set. On the top, there's basically mice embryos sketched, going from a state which is called embryonic state 11.5 to an embryonic state 17.5. And these are sort of course, happening in the embryo, and the neural development starts at this stage 11.5, and by 17.5, it is thought that the early neural development is done. Of course, neural development continues after the baby is born and as it goes on and on, but this is the part where actually the neural tissue itself is established. And the little red square shows you the embryonic cortex, which is where they derive the neural tissue. And then now the pipeline is quite clear. If you work with this kind of technology, you take these cells and put them through a single cell analysis slash sequencing machine. What it gives you is basically it takes each cells and measures the level of transcripts, which is the mRNA copies of various genes that are expressed in individual cells. And in the middle schematic, basically, this is the way which people have sort of come about to visualize this data, where you see these various blobs of colors, and each color blob, each dot on that is a single cell, and each color is basically cells which look more like each other than any other cell in the population. So they are called a cluster, and they have come to define cell types in tissues and across pathologies and whatnot. So now our goal was basically how can we learn metabolism from this kind of data set. So the first thing we found, first idea we had, was that in fact, if metabolism is intertwined with development, which is in this case defined by the new cell types that are emerging, can we sort of ask that question in a more concrete and a rigorous way? So in panel A, in this plot, you can see three colors, which is basically defining the three main cell types in early neural tissue in embryonic cortex. And so these are called, of course, the green is the big one, a big cluster with many cells. Those are the new differentiated neurons. RPs here signifies the progenitor cells. These are like neural stem cells. And then there is a population called IPs, which is sort of intermediate progenitors. They are thought to allow the stem cells to transition to neurons. So then with sort of colleagues at Elucidata, Shashank and Saksham, who were sort of the driving force behind much of the analysis, we basically asked a simple question that if we threw away all the other genes and only keep metabolic genes as defined by, for example, the KEGG database, and we again performed this analysis of assigning cells to different clusters, what is the answer? And it turns out that you can again sort of ask these various machine learning clustering tools to give us three clusters, so three groupings of cells. So here they are shown as S0, S1, and S2, green, blue, and red. And so now the question was, well, there are these two different ways of clustering these cells into three different groups. Do these groups align? In one of the group, again, we use all the genes, all the signaling molecules, all the signaling proteins, the receptors, all the important morphogens, etc. And on the other, panel B, it's only metabolic genes. And what was a big surprise was for us, and also sort of validated our hypothesis, is panel C, which shows that the two ways of grouping cells aligns very well with each other. For example, you can see that RPs, all of the RPs are basically, or 90% of the RPs are falling in state S0. And most of the IPs are in S1, and neuron seems to be a mix of state S2 and S1, suggesting again this sort of transition from RPs to IPs to neurons. So now that we had a good base to stand on, which basically says two things. One is that it is a data set we can believe, because that is again a problem one needs to be very mindful of when working with these high throughput data sets, and at the same time, you have metabolism and cell states aligning very well. So then we sort of went on and performed the simplest analysis one could do and ask, what pathways differentiate RPs and neurons, now that we know metabolism is aligned very well with it. And I should mention here that that this analysis is basically your standard GSEA analysis, and you can start to see in panel D how different pathways are more enriched in RPs versus neurons. And in this plot, basically all the yellow/orange dots are upregulated or enriched, if you will, in RPs, and all the greens dots signify pathways which are enriched in neurons. And of course, as you go towards the right of the plot, the p-value increases and the enrichment increases, and you can start to see various metabolic pathways showing up, but also sort of things like cell cycle and DNA replication being much more enriched in RPs, while exon guidance and endocytosis being sort of more enriched in neurons. So here is panel D sort of enlarged, so you can look at these enriched pathways as well. So clearly, sort of metabolic pathways come up in this enrichment analysis. But however, knowing that fatty acid metabolism is enriched doesn't immediately tell us how and what genes and metabolites may actually be used to control cellular differentiation, which was sort of our a priori goal. Because there are hundreds of genes, and perhaps 10 times more metabolites which might be part of fatty acid metabolism. And also many genes fall into different pathways, so it's not obvious, do we target those genes? Would they have more sort of weight in terms of controlling cellular differentiation or not? So this led to sort of us down the path of trying to establish how we can actually find these underlying metabolites and genes. So here, basically, we developed, used an old sort of tool which Abhishek, the other presenter and one of the co-founders of Elucidata, sort of developed, which they called CombiT. And now it has been sort of put together with many other omics approaches, which have been integrated into a platform called Entomics. And so we sort of used this integrated pathway while developing sort of new ways of integrating and also leveraging single cell data, which has not been done so far. And sort of applying tools of Entomics and the new things we have sort of new ideas we have developed. New ideas we have developed to leverage single cell data, we basically can now make these very detailed maps. These maps sort of lay out not only pick out the important pathways, as you can see, fatty acid metabolism, glutathione metabolism, methionine metabolism, and so on and so forth, but they also pinpoint now the important genes which are basically forming the core of these metabolic networks. And in a nutshell, the primary thing we leverage here is that we actually know all the reactions and reactants very well in metabolism. So we can actually build these networks based on knowing only the genes and not knowing at this stage the metabolites. So it sort of generates lots of new hypotheses on which metabolites might be important and allow us to make progress. So here I would like to, for example, highlight one of the things which comes out also is pathways like glutathione metabolism are not picked up by these global analyses such as GSEA, partly because there are only handful of genes which are sort of differentially regulated between the two cell types. However, when you put them together in a network, it becomes very clear that they are essential in defining the differences between RPs and neurons. And the goal again is, now that we have a much high resolution view of how metabolism is rewiring as RPs, which are the neural stem cells, are forming neurons. Now we can even perhaps look for therapeutic targets which can alter neural developments in case of pathologies. Again, and the power of the data set we were working with is that it actually had samples from different time points. So not only a single snapshot, but it had data sets from multiple time points. And here we show the analysis for 3 time points, which is 30, embryonic stage 13, 15, and 17. And here we could immediately see, so this is the top left panel, which again, we went back to the GSEA analysis and shows that one of the most important change with time was the fatty acid metabolism, which was getting enriched rapidly as these RPs were changing in age from 13 to 17. However, we could find that, as shown in the Venn diagram right below that, is that there is a core set of 29 genes which sort of remained constant across these 3 time points, while they were sort of various subset of genes which are only important at different time points. So to make it much, to look at it at a finer resolution, we again went back to making these metabolic networks. And now we take only the RPs at 2 different time points and start making these metabolic networks. And of course, much of the analysis replicates what GSEA does, but at the same time, it sort of brings out various new elements to it. And one of the things which I would like to highlight is basically the upregulation of mevalonate and cholesterol biosynthetic pathways. And this is sort of a characteristic pathway which comes up in these old RPs and has not been seen sort of anywhere else so far. On the other hand, we see that the methionine metabolism, which you can see on sort of lots of genes which are looking green, are actually down-regulated as RPs transition from day 13 to day 17. And in some ways, they're shutting down the methionine metabolism and upregulating their cholesterol biosynthesis, which is sort of tied in very closely with the fatty acid metabolism. So now that we had a good view of how RPs are changing with time and what RPs and neurons sort of differ metabolically, we also wanted to ask whether our findings sort of remain consistent if we go to a different region of the developing brain. And in this case, we basically leveraged data which was publicly available, and something which is published and not generated locally, to sort of challenge our analysis and cross-check our findings. And so this is a data set from a different region of the brain called dentate gyrus, and this data was extracted at embryonic stage 16.5. So right close to the endpoint, which was the 17.5 in the previous analysis. And again, to our sort of surprise, but at the same time sort of satisfaction, that important pathways such as mevalonate and cholesterol biosynthetic pathways were also upregulated at this late stage in dentate gyrus, which sort of, along with sort of consistency across all the other pathways. And this figure may be a little bit hard to read, but all the dotted reactions, the dashed lines, are basically common reactions across everything, across the two analyses. So that gives us a lot of confidence in our analysis, and we can sort of perhaps pursue some of the hypotheses and therapeutic targets which are sort of highlighted by this analysis. And here is a slide to sort of highlight a few of them. So here I've sort of zoomed into our metabolic network, which we have assembled. For example, around acetyl-CoA, which is basically an important epigenetic modulator. And here, for example, we have highlighted several genes. So some of them are marked blue. So these were sort of, we are suggesting as possible targets, which we can target to change the fate of cellular differentiation by changing the way how the neural stem cells, which are called RPs, are affected. While at the same time, sort of, we highlight some of these genes in sort of greenish color, which are the genes which have already been identified and are targeted in literature to control neural development during various pathologies. So this brings me to conclusions. So as we said, as Elucidata now has, is a mix of lots of tools, transcriptomics tools put together, omics tools put together, which they call entomics. It sort of shows, confirms pathways and genes which have been found in earlier data. Many of them were in vitro. And this new analysis now points towards genetic and metabolic drivers, which might be sort of tweaked to control the epigenetic machinery, which are important for sort of maintaining pluripotency of stem cells and also how they differentiate in time. The third big point was we actually also identified various pathways such as mevalonate and cholesterol biosynthetic pathway, which upregulates as cells sort of slow down, as neural stem cells slow down and become quiescent, if you will, as the neural tissue reaches this early development, ends its early development stage. And again, to highlight things like glutathione metabolism, which have a strong role to play in quiescence and neural development, will sort of not be really picked up with sort of global analysis such as GSEA, but comes up immediately in the kind of analysis we have developed together. And finally, it sort of provides new targets, both metabolic and genetic, which could then be addressed. Okay, with that, I hand over to Abhishek. Thank you.
Hear me fine. Great. So just in the interest of full disclosure, like I have known Sidharth for like around 2 decades now, and it has been a really exciting and a lot of fun to work with him over the last couple of years in this context. And this collaboration has been a huge learning experience for us. Sidharth shared a bunch of biological metabolomic insights that came out of this collaboration. What I will try to do in this second half of the presentation is walk you through the other side of the same story as to what enabled this. And clearly, for any one of you in the audience who is interested or has used single cell RNA-seq data set, you'd be aware of a lot of challenges. At the same time, the promise of this data set is also immense. There's a snapshot of what you see on the left of all the publicly available data sets, single cell RNA-seq data sets on Geo today. And many labs focused on very different aspects of biology are leveraging this new technology. And just in the last few years, it has completely change how the resolution at which we can look at the biology at hand. And we showed you some examples of how using two publicly available data sets, we could do that with SIDS Lab. And as I said, what I'd like to do now is to look at the same story from another perspective as to what are the underlying challenges, what makes it harder for scientists at large to do that over and over again. So again, in a very simplistic world, if you were to think of two extreme personas, scientists, you'd identify yourself either as a biologist, bench scientist, or a bioinformatics scientist. Clearly, it's a continuum spectrum, and there are many biologists who can code and many bioinformatics scientists who know their kinases really well. But you'd identify with these pain points. You must have asked these at some point in the last few months, where you did find a paper which has a very compelling single cell data set, but a lot of us don't even know how to access it to begin with. And some of you might have used a core facility in your university or a CRO that you like to get data generated on a system of interest, and that delivery of the data set in terms of gigabytes of information and a PDF report which is static it leaves you hanging as to what does that actually mean? How does that drive my discovery effort? And that has been very limiting. And eventually, it's been very consistent in our experience, much like in this collaboration, that there are many stakeholders involved in trying to extract insight of this data set. And how do you collaborate and how do you share the data, the parameters, the Python notebooks with your colleagues? That has been a very challenging aspect of any such endeavor. Now, if you are a bioinformatics scientist, a lot of us in this audience, I hope, fit that description. They would be very comfortable with writing a Python notebook to do the analysis, have their own custom workflow, which is an essential part of any such inquiry. But often these data sets are so large that you cannot possibly run that on local system. It would be very painful and hard and slow. Well, in that case, you look up to some cloud infrastructure, either AWS or GCP or Azure or your own university local, high performance computing clusters. And that introduces a barrier. It's surmountable by some of us, not a lot of us, but it's a bottleneck nonetheless. Now, in many cases, as a bioinformatics scientist, you might also run into a paper that you want to reproduce, and that is challenging. It can anywhere take you from a week to many weeks because the data is not standardized, the underlying analysis and the code is not available in the most readily form. And we experienced some of that too, and we had to reach out to the authors to figure out those details. But a bottleneck nonetheless. Then again, there's a huge number of tools that keep coming up from many academic labs almost on a weekly basis that help you analyze the data, process the data, visualize it. And it's always a question as to which tool is the most relevant one, and that requires a fair bit of homework to figure out the right answer. And as you have a processed data set, your Disney maps and whatnot, you want to figure out what are the most relevant markers, and the publicly available list of markers for immune cells or other kind of cells can leave you wanting in some cases. So again, there's a need, a pain point that we have seen over and over again, where a biologist or a scientist would want to have their own list of markers for that particular biological system of interest. And sharing again from this perspective too is a challenge. Like, how do you share the plots that you have generated in your Python notebook on a slide deck in a group meeting? at a regular basis to get feedback from your biologist colleagues and keep driving the process. Again, it's almost a laundry list of pain points that we have experienced ourselves, and we have heard over and over again with our customers that are using single-cell RNA-seq data to drive their discovery efforts. And I hope and believe that a lot of this would resonate with a number of folks in this audience today. And this essentially defines as to what we are trying to address as a company. And Poly is the platform that we have built. It's on the cloud. It's cloud neutral. It uses both all three of the major cloud providers, AWS, GCP, and Azure. And specifically for this context, we'll have a closer look into this. But what it does for scientists and the community of scientists at large is it enables democratization of data. If you're a computational biologist or a very mathematically inclined biologist, it can also help you simplify and scale analysis. And finally, the whole endeavor has been done in the first place to extract some insights, and Poly enables you with that too. Again, we saw an example with Sid walked us through, and there we looked at only 2 publicly available data sets. There are hundreds and thousands of them, and how can we do that at scale? Poly helps its users do that. So let's look at the first one, where we are enabling democratization of data, that is for the scientific community. And again, we have seen this, the pain points have been very consistent. It doesn't matter if you're an academic lab or a small biotech or a large pharmaceutical company. Single cell data is undoubtedly large and hard to store and manage. And often you also rely on publicly available data sets, much like how we did in this particular first half of the presentation, which will soon be a manuscript available on bioRxiv. Publicly available data sets are hard to access, even though they are available not behind a password or paywall. It's hard to use them as in a meaningful way. And partly that is accentuated, that challenge is because of lack of standardization across data sources and analytic pipelines. So what we have done on Poly to enable scientists like you is to create an infrastructure that securely stores and manages your data sets and different versions of it without really worrying about too much about the infrastructure. And that frees you up to focus more on extracting insights to drive your discovery efforts. We have taken a number of publicly available data sets, and Shashank, my colleague, will give you a live a demo of how this looks like in action after I'm done with a few of my slides. All of the 3 main components, the data, code, and analysis report, are available on one single platform in Poly. And often in practice, by the time you're done with this data set, much like what we did with this 2 publicly available data set, you end up with a number of versions of data sets. And most simplistically, in some cases, you remove a sample, in some cases, you add 2 data sets, and so on and so forth. The number of versions and managing them is a challenge. Similarly, you end up with multiple notebooks, Python or R scripts or whatever you may want to use, and multiple versions of them. And finally, analysis too. Like all of us who have written a paper, we know that we always have this underscore final and final and final. So there are many versions of analysis too, which are often scattered all over, and Poly puts them all on one place for centralized access to you and your colleagues, which again introduces a number of efficiencies. And all of this can be shared seamlessly across the team. Poly Data Lake is the outcome of this endeavor, where we have put 400 data sets, all single-cell RNA-seq, all publicly available, curated them, standardized them, annotated them, the data as well as the metadata, and it's ready to use. It's also available through programmatic access through our APIs, and we would be very happy to have any conversation around that. You can take the raw data, the publicly available data set, push them through your processing pipeline to have a standardized data set to compare them across different experiments. We have our own processing pipeline, which we'll be also happy to make available, which is what we used in this work that Sid talked about earlier. And this will be the main focus of the demo that you will see towards the end of this presentation. Again, focusing on the analysis part, single cell analysis is in some ways uniquely complex, definitely more so compared to bulk RNA-seq analysis. The dimensionality of the data is huge for the obvious reason that you have readout on almost every single cell. The annotation and other more quote-unquote standardized downstream analysis of bulk RNA-seq do not hold here very well. So that poses a different kind of challenge for a computational biologist. And again, Poly enables or aims to serve and address those pain points as well. So in a nutshell, you can just focus and worry about your Python notebook and not worry too much about the cloud infrastructure, associated cost, or any management overhead thereof. So you can just stay focused on your Python notebook or your R script that you would have. All the data, code, the environment, and the analysis reports, and different versions of all of them live in one place, which is available to you and your team. Again, in a very high level, the schematic of how it works is you have Poly workspaces. So, for example, this would be the project that Sid talked to you about would be a workspace on Poly where you can bring and manage data, code, and environment, and share it with your colleagues. You could use notebooks that are pre-configured or have your own notebook, Python notebook put on Poly so that it reflects exactly the way data should be processed per your needs. You can also use command line interface of Poly to process the data. If you are the kind of person who does not want to click many things and you want to use your keyboard and screen to just run a number of analysis on the cloud without a user interface. You can monitor tasks and therefore the cost of the cloud computation. There's a detailed log that is available, and there's a lot of automation inbuilt to manage the costs associated of single cell RNA-seq dataset, which can creep up very fast if you're not very careful. So we have, again, aimed to minimize the overhead of managing the infrastructure, the cloud infrastructure for a computational biologist who's focused mostly on extracting insight and processing the data set in the best possible way. Eventually, like sharing, again, is a critical aspect of how we do things, and we enable sharing of analysis of the reports and the notebooks and the data as you deem appropriate with the rest of your colleagues. Enabling insights, again, that is the end game here. That's why you do any experiment to begin with. Again, here there are some consistent themes that come across where for a biologist who has a lot of context as to why this experiment was done in the first place, they would like to compare a particular gene across different types, cell types, and across different data sets, public and proprietary. You'd want to generate some actionable hypothesis for whatever biological context that you would be interested in using these experiments, these data sets. And again, the way we have tried to approach that is to build very intuitive dashboards, which are highly customizable for your needs and the questions that you're interested in. We have made available some of our proprietary tools. Intermix is one of them that Sid talked about. There are other machine learning modules that we have built and made available, which helps you extract insights from both public and proprietary data. And finally, for any of these analysis, you can generate a one-click report, which you can share with your colleagues across the lab or across the ocean. With that, I'll pause and I'll ask my colleague Shashank to walk us through a demo of Poly and to show you how the single cell capabilities look like in action. Thanks.
So we'll be showing you a video since our internet connections are a little bit spooky. So we'll talk about Yeah, so basically, this is what Poly Data Lake looks like. It contains things from Geo, AML, GBM, transcriptomic datasets, single cell RNA-seq datasets, and also COVID-19 datasets. So we are allowing researchers free access to COVID-19 datasets. So if you want the access, do let us know. So basically, each of the data lake or data repository consists of basically an overview dashboard. Here you see an overview dashboard, and you see different cell types representing this single cell data lake, which organisms they belong to, which disease do they belong to, how many data sets are there, and also like which tissue uh, do they correspond to as well. You can also basically subset the datasets on the fly. And you can apply filters in the overview dashboard to see the subset of datasets. And then you can also, like, we provide custom dashboards for exploring these datasets. So here you can see one of our custom dashboards. And these are highly customizable. We can get your own workflow in here as well. So you can see different kinds of options for single cell visualization. One is our favorite, finding our favorite gene. So here, basically, we can get every gene expression across cell types and datasets. So here you can see the PTPRZ1 gene that is upregulated in astrocytes. It's basically a marker, and it also came up in Intomics results as well. Uh, so yeah, so basically this is how you can in real time see data across data sets and see if your marker holds value or not. There's another marker which people use for neurons. Uh, you can see that this is highly upregulated in neurons and fibroblasts as well. And you can also see the data sets in which these are going up, which I'll come to later, that you can actually go into that data set as well. So here you can basically search by keywords. You can search in different fields as well. And right now we have 400 data sets, publicly available data sets, which have been curated. Yeah, so here we search for glial, we get these many data sets. And all of these data sets have their corresponding GeoSeries ID and their publication PMC IDs as well, so that you can directly revert back to the actual publication as well. So here, basically, we search for glia, and yeah, you can see organism and publication as well. And you can also see the paper that Sidharth talked about earlier. So we'll select one of the data set and look at it more closely. Uh, so basically, it involves just clicking that data set and loading it up. So it will download that data set not on your own computer, but on the server, and will expose the complete data set to you. So once that is done, basically you can see quality checks, variable genes, and other kinds of TSNE visualization as well. So in the quality check matrix, you can see uh, gene counts, UMI counts versus the cluster type, and also like the mitochondrial gene expression, which is a way to filter out dead cells or even doublets as well. So in the variable gene, you can see which genes might be driving it. These are your most predictive biomarkers. Here you can see UMAP and TSNE plots. You can also select the curated cell identity and basically see which cell types do these correspond to different kinds of cells. And you can also search for different genes and see whether they are your markers in a particular data set or not. So this is a more granular way of doing things rather than the first plot that we showed you earlier, which gave you a more global thing. So here you can see that TUB3 is more expressed in neurons through different kinds of plots, like violin and dot plot. Uh, a very special capability we have here is that you can also see multiple data sets together. Like I can select Siddharth's data set as well and see both of them together. Uh, basically, once you do that, then you can go through the same stream. And basically, you can select datasets here to switch between datasets and get the other datasets quality check metrics like mitochondrial content and the gene counts, the UMI counts plot, which is very good in this dataset. And you can also see variable genes as well. Um, and if you remember the earlier slide that Siddharth showed, uh, we'll see that almost similar kind of TSNE plot we get for RPs, IPs, and neurons. You can see on the right hand side, and so you can reproduce it real time. And then you can also see the top 3 expression, which is uh, more in neurons. So it is basically uh, more in both the data sets that we have seen. Yeah, I think that's about it. This was a quick video. So now we are open for questions. So we'll take up questions in a bit. Yeah. So you can type in your questions in the chat box that is present on the right hand side. We are eagerly waiting for your questions, by the way. Yeah.
Okay. Can I? From the audience, which is, and I hope you can respond to this. Can you query multiple genes at the same time?
We can query multiple genes. So in the marker section, we can actually query multiple genes at the same time and see them. Yeah.
That I'm reading, I'm interested in understanding differentiation of muscle cells. Do you have public data set? So I can respond to that, and Shashank, please feel free to chime in. So my answer would be most likely yes. Of the 400 plus data sets that we have curated, there must be some that would point to differentiation in muscle cells. If not, if you're interested in if you know of a particular paper, particular data sets that are not part of our data lake, we'd be happy to add them to the data lake. But one of the things that Shashank was showing is there's a very simple query interface where you can look for different keywords like muscle differentiation and see what data sets are available and probe those data sets even further. Shashank, do you want to add to that?
We do have muscle cells as well, and you can also search by cell types as well. So you can get those data sets pretty quickly. Yeah.
Wonderful.
And yeah, you can also analyze the data generated in your lab as well on Jupyter notebooks and then visualize them through these apps, which you can share among your colleagues.
Wonderful. Thanks, Shashank. The next question is from Sarat. I hope I'm pronouncing your name right. How is the data cleaning done on all different data sets? And again, I'll briefly comment on this and I'll have Shashank get into details. We try to start from scratch and have the data processed at different stages available so that they can be fed into your own custom pipeline for comparison across different data sets, if that is a question, if that is something of interest. Right now, the cleaning is focused on one data set at a time, but we also have annotated and standardized the metadata across different experiments so that the query or the comparison across different data sets is enabled. I'll pause here and I'll ask Shashank to add more to that. And correct me if I'm wrong, if I was wrong.
Yeah, so basically, all of these, the good thing about single cell datasets is you can find the raw data submitted in Geo or any other repository, and you can basically run your own pipeline on it. So we have run a star pipeline and cell ranger pipeline on these, and basically got the counts for these data sets. And for the metadata, we have basically NLP algorithm which can identify which cell types do these data sets correspond to, and based on that input and the data input, we and the manual curation, of course, we manually curate the cell types present in the data sets. And that is how we do it right now.
Thanks, Shashank. The next question is from Aram. Hi, Aram. The question is, are the number of public experiments available in platform added regularly, or is these the 400 the base set? So again, I'll chime in, and then Shashank can add to this too. So these data sets are updated regularly. They're every once in a while we update this, and the proprietary tool that we have developed it makes that updating very easy for a scale of data set that we're dealing with here. The 400 data set is the base set right now, but we'll keep updating this on a monthly basis going forward. Most of them are sourced from a resource that has been created by a group at Avi Mayan's lab at Mount Sinai. But again, I'll pause here and let Shashank add to the comments.
Check on current COVID-19 situation as well. Basically, we are trying to extract those data sets from bioRxiv and other places as well. And yeah, we monthly review the number of data sets. So it is not at all going to be 400. So this is going to grow. Yeah.
What we have started with to release the product. Next question is, I'll read out the question. Is it possible to create a data lake from our in-house data sets? So I don't know who is the person. I don't have a name associated with this. The short answer is yes. Often the use case of Poly is where labs, both academic and industrial, use a combination of public data sets with their own proprietary in-house data sets. Of course, the volume is dominated by what is publicly available, and they have a steady stream of one data set per month kind of frequency to add their proprietary data set to this data lake. So yes, that can be done, and it is how it is in use in most of the cases. Another question that I have is, are larger data sets generated using the 10x genomics platform available? Shashank, do you want to respond to that?
Other data sets available from 10x Genomics and other platforms as well. So some of the larger data sets like Brain Mouse Atlas and other data sets like Endothelial Atlas are also, so we are trying to make them available on the platform as well. But we do have data sets which are more than 300 or 400 gigabytes in size on the platform as well.
A question from Amita is this organism specific? And I'll again comment on this, and Shashank, feel free to add. So the short answer is no. If you, one of the first dashboards that Shashank showed you in the video was where you can look at different organisms that contribute to this 400 plus data sets. And so you'd find all those things that are available in the public domain. If there's a particular organism that you're interested in, or a particular data set or database that is of interest, please feel free to reach out to us. We'll be happy to add that to the data lake. But again, I'll wait and pause and hand it to Shashank.
At all, so we can put any organism or data into it. So this is just a way of basically collecting your data and putting it in an organized fashion. So this is what the data lake is about. Yeah.
Thanks, Shashank. We have another question from Sarath. Thanks, Sarath, for asking multiple questions. keep them coming. The question is, can poly be used for bacterial cell data, or is it limited to eukaryotic cells, mouse, human only? Again, I think this touches upon the last question that we addressed from Amita. There are no such constraints. It can be used to look at bacterial cell data too. Shashank, do you want to add more to this?
Is very interesting because you can also basically see metabolism in those cells as well. So yeah, poly can definitely be used for bacterial cell data, and you can use Entomics to derive metabolic insights from it
Thanks,
as well.
Shashank.
Yeah.
Next question again from Sarath. Really like this. Question is, any plans of using gene interaction and regulation networks to reduce noise from single-cell RNA-seq data? Shashank, feel free to comment on it. My initial thoughts would be that the example that Sid walked us through was pretty much using the same approach in a sense, where we were using metabolomic network as defined by KEGG to reduce noise and extract some signal out of single-cell RNA-seq dataset. We do have plans to integrate this with other gene interaction and regulation networks that are publicly available. But again, I'll wait for Shashank. And Sid, feel free to comment on this as well.
Yeah.
Shashank, do you want to go first?
No, I'll ask it too. Yeah.
Okay, so I guess I'm assuming you can hear me. Make sure, yeah. Okay, good. So I think the short answer is beyond the metabolic part, which we showed, where we, as Abhishek said, we really leverage the gene interaction and the known network to sort of really reduce noise and bring out things like glutathione metabolism, which were sort of lost in noise when you look at global analysis. But a similar trick one could perhaps play with, for example, signaling networks. The challenge there, of course, is that we actually don't know who talks to who. That is really the biological challenge there. And people sort of continuously are trying to map it. And not only is it sort of we don't know who talks to who, these players keep changing from context, even within the same organism, right? So different tissues might have different interactions going on. But again, sort of, I think in terms of speaking technically, the tricks we use to understand metabolism, if you have a known network, same can be applied for any network, actually. Yeah.
Right. And just to add to Sid's comment, this is exactly what we see as an advantage of working with metabolomic networks, because they are, relatively speaking, better defined and better characterized than, let's say, signaling networks. But in principle, the approach would be applicable to any network as such. Another question is, if I want to do other types of analysis, like trajectory analysis, can you add that to the single cell screen? Can it be customized for our experiments specifically? So I suppose this question is referring to the dashboards that Shashank was showing in his video. Again, my initial comments would be yes. The dashboards are highly configurable, and they can be made to your custom specifications. We can do that for you. You can do that yourself as well. Again, I'll hand it over to Shashank for a deeper answer.
This dashboard, this is just an example of dashboard that we showed you. So basically, it's not like that if an analysis is inside this dashboard, it will take months to put another analysis in it. So we do have a track record of making an analysis available within a week, which is used in your own lab. So yeah, so we can add any kind of analysis to the single cell. Yeah.
Wonderful. So now we're at the top of the hour. I want to thank everyone for participating, taking an hour out of your lives to come and join us. These are surreal times, so please be safe and exercise caution as advised by whoever you trust. Be safe, and again, we are very thankful that you took time to join us. Thank you, everyone.